Welcome to today's lecture on the ABO blood group system, which remains the most important system in both transfusion and transplant medicine. While we often take it for granted, the discovery of this system by Karl Steiner in 1900s and the subsequent addition of Group AB by his associates in 1902 marked the true beginning of modern blood banking. The cornerstone of this system is Landsteiner's rule or law, which establishes that normal, healthy individuals predictably possess ABO antibodies in their plasma directed against the specific ABO antigens that are absent from their own red cells. This creates a unique inverse reciprocal relationship between the antigens we find on the red cell surface and the antibodies circulating in the serum. Why do prioritize ABO above all other systems? It is because these antibodies are naturally occurring or non-red cell stimulated, likely developing from environmental exposure to similar structures in nature, such as bacteria and pollen. Consequently, if a patient receives ABO incompatible blood, these pre-existing antibodies can cause immediate intravascular hemolysis, leading to a severe and potentially fatal acute hemolytic transfusion reaction. Biologically, these antigens are quite fascinating. Uh, First, because they aren't just on the red cells, they are found on platelets, lymphocytes, epithelial cells, and most organs like the kidneys. Next is that they are detectable as early as 5 to 6 weeks in the utero, though they do not reach full adult levels of expression until a child is between 2 and 4 years of age. So the production of these antigens is a complex process involving the interaction of the ABO, big H, small h, and the SE genes. Um, Everything builds upon the H antigen, which serves as the mandatory precursor structure. So the specific type is determined by transferase enzymes adding specific sugars to that H base, the N-acetylgalactosamine for group A and D-galactose for group B. So in this lecture, we will dive deeper into the genetics on chromosome 9, different subgroups like A1 and A2, which will be discussed on a different period, and the essential laboratory techniques used for forward and reverse gripping to ensure every transfusion is a safe one. So the ABO system is unique because individuals naturally have antibodies against antigens they lack without prior exposure to red cells. So transfusion of an incompatible ABO type can cause immediate lysis of donor RBCs, leading to a severe and possibly fatal transfusion reaction. So the ABO blood group system forms the foundation of all pre-transfusion testing. Now we have here this image. This image represents the red blood cell membrane structure, highlighting key proteins, glycoproteins, and carbohydrates involved in blood group antigen expression. So we have here the lipid bilayer and the embedded proteins. So the lipid bilayer forms the main structure of the RBC membrane, providing flexibility and integrity. So we also have glycophorin A and the glycophorin B, which are integral membrane proteins that help in the expression of the blood group antigens, including the MNS blood group system. We also have here the ABO blood group system and as well as the carbohydrates. So the ABO antigens such as A, B, and H antigens are carbohydrate structures attached to a glycoprotein and glycolipid on the RBC membrane. So these carbohydrates are present on the extracellular surface as seen in the diagram, labeled as carbohydrates, close and open parenthesis ABO, and as well as close and open parenthesis Lewis. So the ABO system is determined by the type of terminal sugar. A antigen has N-acetylgalactosamine, B antigen has the D-galactose, O group lacks these sugars but has the precursor H antigen. So the role of spectrin and cytoskeleton in the blood cell shape, spectrin tetramer actin, and as well as ankyrin are cytoskeletal proteins that maintain RBC shape and deformability. So while they are not directly related to the ABO system, they influence RBC survival and flexibility, impacting how antigens are displayed on the cell surface. So we also have here in this image the RH system, another blood group antigen. So the RH glycoprotein and RH polypeptide are transmembrane proteins that play a role in RH blood group antigen expression, such as RHD, which determines if a person is RH positive or RH negative. We also have the band tree here, in which you can find the Diego blood group antigen. It is also another key transmembrane protein important for ion exchange. So the importance of this structure in transfusion medicine. So the ABO system is the most critical for blood transfusion because incompatible transfusions will lead to severe hemolytic reactions. Since the ABO antigens are present on all RBCs, their proper identification is crucial before any transfusion. So we have here ISBT, or the International Society of Blood Transfusion. So it's an international scientific society for transfusion medicine professionals founded in 1935. So while it is not a regulatory or accrediting body, its primary mission is to improve the safety of blood transfusion on a global scale. So according to our references, the ISBT's work is organized through several key functions. So the first is for universal nomenclature and classification. Next is to standardize numeric terminology. Third is using the ISBT 1 to 8 labeling standard, in which it provides a global standard for the identification and labeling of medical products of human origin, and that is known as your ISBT 128. Okay? So, due to inconsistency of blood group antigen terminology, so that's why ISBT created a working party for standardization of your red cell antigens and blood group system terminologies way back in 1980s. So next, the discovery of Sir Karl Landsteiner marked the beginning of modern blood banking and transfusion medicine. So Karl Landsteiner's contributions are the foundation of modern blood banking, most notably his 1900 discovery of the ABO blood group system. Through experiments mixing random human blood samples, he recognized distinct patterns of clumping known as agglutination. So based on these observations, he originally described blood groups A, B, and O, together with his associates adding group AB shortly after. So Landsteiner's most significant finding is Landsteiner's rule, or Landsteiner's law. So this rule establishes that normal healthy individuals possess ABO antibodies in their plasma directed against the specific ABO antigens that are absent from their own red cells. So this inverse relationship is unique to the ABO system, and it is essential for determining transfusion compatibility. So again, Karl Landsteiner discovered the ABO system in 1901, laying the groundwork for modern blood banking. So the clinical importance of Landsteiner's work includes ensuring transfusion safety, and it is also the foundation of compatibility, and as well as the discovery of the RH system, wherein way back in 1940s, Sir Karl Landsteiner collaborated with Alexander Weiner to report the discovery of the RH blood group system. So again, Landsteiner law, also known as the Landsteiner's rule, is the fundamental principle of the ABO blood group system, establishing that a person's serum will always contain a predictable antibody, or will always contain predictable antibodies directed against the specific ABO antigens that are absent from their own red blood cells. So discovered by Sir Karl Landsteiner in 1900s. So this unique reciprocal relationship again is the foundation of modern blood banking and transfusion safety. So this image right here shows the rule the Landsteiner's rule in practice. So according to this law, the antigens present on your red cells dictate which antibodies will be in your plasma. The relationship for the four major blood groups is as follows. For group A, these individuals have the A antigen on the red cells because they lack the B antigen. That's why they possess anti-B antibodies in their serum. Group B, these individuals have the B antigen on the red cells because they lack the A antigen. They possess anti-A antibodies in their serum. Group AB, these individuals have both A and B antigens on the red cells. Since they lack neither, they have no ABO antibodies in their serum. Group O, these individuals have neither A nor B antigens on the red cells because they lack both. That's why they possess both anti-A and anti-B antibodies in their serum. So while these antibodies were traditionally called naturally occurring, our references clarify that they're actually non-red blood cell stimulated. So the ABO antibodies could be stimulated by the environment, and maybe it could be also related to immune response, and sometimes it could also be related to developmental timing, wherein these antibodies are generally not detectable until an infant is 3 to 6 months old. So blood typing, also known as the blood grouping, is the process of identifying specific antigens on the surface of the red blood cells and the predictable antibodies circulating in the plasma. So it is the most frequent and critical test performed in a blood bank because transfusing incompatible blood can trigger an immune response leading to immediate, potentially fatal intravascular hemolysis. So for the ABO blood group system, the ABO system is the foundation of blood typing because it is the only system where antibodies are predictably present even without prior exposure to foreign blood. So typing for ABO involves two distinct tests that must match. So we have the forward grouping or the front typing, wherein the medical technologists use known commercial anti-sera such as anti-A and anti-B to detect which antigens are on the patient's red cells. For example, if a patient's cells clump when mixed with anti-A but not anti-B, they have the A antigen. While reverse grouping or the back typing, this confirms the forward type by using known reagent red cells such as A1 cells and B cells in order to detect antibodies in the patient's serum. And remember, always remember the Landsteiner's rule. This rule establishes that normal individuals possess antibodies to the ABO antigens they lack on their own cells. So for instance, a group A individual will have anti-B antibodies in their serum. So this table shows the frequency of ABO blood groups among selected populations and ethnic groups. So it shows here that the ABO phenotype frequencies in the United States vary significantly depending on the race and ethnicity. So across all major ethnic groups, Group O is the most common phenotype, while Group AB is consistently the rarest. So these variations are the result of diverse blood group gene inheritance patterns with specific populations. So for the white population, the most frequent type is Group O at 40%, followed closely by 45%, followed closely by Group A at 40%. Group B accounts for 11%, and Group AB is the least frequent at 4%. For the Black or African American population, this group has a higher prevalence of Group O, usually at 49%. Notably, the Group B phenotype is significantly more common in this population, usually 20% than in the white population. So Group A occurs in 27% of individuals, and Group AB remains at 4%. For Asian population, this group has a more evenly distributed frequency of Group A at 28% and Group B at 27%. Group O accounts for 40% and Group AB is more frequent in this population at 5% than in other US ethnic groups. So for according to the Department of Health here in the Philippines, the most common blood type is blood group O, followed by A, then B, and AB. So more than 99% of the Filipinos are Rh positive, while less than 1% has the Rh negative blood. Now let's talk about the ABO antibodies. So they are naturally occurring because they are produced without any exposure to RBCs. So in small quantities of IgG may also be present, but specifically for anti-AB, it's not a combination of A and B but totally different antibody. So ABO antibodies are otherwise known as your isoagglutinins. These are the most important antibodies in transfusion medicine because they are predictably present in the serum of a normal, healthy individual. These antibodies follow Landsteiner's rule, which states that individuals possess antibodies directed against specific ABO antigens that are absent from their own red blood cells. For example, a group A person lack the B antigen and will therefore have anti-B antibodies in their plasma. So, for the origin of these ABO antibodies, again, I have already mentioned this one, it could be an environmental stimulus. So, while traditionally called naturally occurring, these antibodies are more accurately described as non-red blood cell stimulated because they develop after exposure to environmental substances like bacteria, pollen, and plants that have chemical structures similar to A and B antigens. So please take note that ABO antibodies are not detectable at birth. So they typically appear when an infant is 3 to 6 months old, so reaching their highest levels between the ages 5 and 10. Take note also that antibody levels naturally decrease in elderly adults, which can sometimes lead to weak or missing reactions during laboratory testing. So as for the type of antibody a person produced depends on their ABO blood group. So let's say for group A and B individuals, their anti-B or anti-A is predominantly of the IgM class, though they may contain small amounts of IgG. I have already mentioned also this one in the previous discussion that I have. So group O individuals, so their anti-A and anti-B are primarily of the IgG class. So this is clinically important because IgG antibodies can cross the placenta and potentially cause hemolytic disease of the fetus and the newborn. For the anti-AB, Group O individuals also possess a unique cross-reacting antibody called anti-AB that can bind to both A and B antigens and cannot be separated into individual anti-A or anti-B components. So the clinical significance of the ABO antibodies, of course, to detect intravascular hemolysis because ABO antibodies are efficient in activating complement. So they also can produce agglutination. So in the laboratory, ABO antibodies react best at room temperature, usually 20 to 24 degrees Celsius or colder. So they are famous for producing strong direct agglutination or clamping during the immediate spin phase of testing. So typically graded as 3 plus or 4 plus, which indicates a strong reaction. Now let's proceed to the inheritance of A, B, and O antigens. So way back in 1924, according to Sir Felix Bernstein, he proposed the multiple allele theory, in which it states that an individual inherits one ABO gene from each parent, and that these two genes determine which ABO antigens are present on the RBC membrane. So it just follows the simple Mendelian genetics. So meaning that an individual inherits one ABO gene from each parent. So these genes are located at specific position or locus on chromosome 9. So the three main alleles. So we have alleles A, B, and O. So these interact in three distinct ways. So let's just review your blood bank genetics. So we have codominant. The A and B genes are equally strong. If an individual inherits an A gene from one parent and a B gene from the other, both antigens are expressed on the red cells, resulting in the AB phenotype. For dominant, both the A and B genes are dominant over the O gene. This means if you have one A gene and one O gene, your blood type will be A. For the recessive, the O gene is an amorph or a silent gene, meaning it does not produce any detectable antigen. So to have type O blood, an individual must inherit an O gene from both parents. So that's why the genotype of an individual having blood group O has the genotype big O and big O. Now let's talk about the precursor substance. Okay? So a precursor substance is a basic material or a base that is converted into another substance by adding a specific constituent such as a sugar residue. So in immunohematology, it serves as the antigen building block upon which blood group markers are constructed. So let's define the following terms. So we have the paragloboside. So it's also known as a glycan. It is the specific precursor substance from which the A, B, and H antigens originate. So it is classified as a type 2 precursor and functions as the primary substrate for the formation of both H antigens and the P1 antigens. Oligosaccharide chains are chemical compounds formed by a small number of simple carbohydrate or sugar molecules. So these chains, which can be linear or branch, serves as the common structure for A, B, and H antigens and must be attached to a protein or lipid carrier molecule to exist on the cell membrane. We also have the glycosyl transferases. These are specialized enzymes that act as biological builders by catalyzing the transfer of glycosyl groups or the simple sugars from one molecule to another during biochemical reactions. So in the ABO system, these enzymes are the primary gene products. They recognize a specific precursor and attach a unique immunodominant sugar to it, which determines the individual's blood type. And lastly, type 2 precursor substance, it refers to a specific chemical linkage within the oligosaccharide chain where the number 1 carbon of D-galactose is attached to the number 4 carbon of N-acetylglucosamine, which will give you a beta 1 to 4 linkage. So this specific structure is the predominant type found on the red blood cell membranes, whereas type 1 structure are primarily associated with body fluids and secretions. So now, let's proceed to the precursor substance. So both type 1 and 2 precursor substances consist of a basic oligosaccharide chain as shown in the image, which includes galactose or the GAL, N-acetylglucosamine or the GLC-NAC, another galactose or the GAL, a protein or a lipid which is represented by R to which the chain is attached. So the key difference lies in how the sugars are linked to each other. Type 1 precursor substance, which is found in body fluids and secretions, has beta 1,3 linkage, in which your galactose is attached to N-acetylglucosamine via a beta 1,3 bond. So, type 1 precursor substance is present in the secretions and body fluids such as the saliva, plasma, etc. So, type 1 precursor substance determines whether a person is a secretor, meaning can express ABO antigens in the body fluids. So type 1 precursor substance is modified by the secretory gene or the FUT2 or the fucosyl-transferase to form soluble A, B, or H antigens. While type 2 precursor substance is found on the red blood cells. So they have the beta-1,4 linkage, which means that galactose is attached to N-acetylglucosamine via a beta-1,4 bond. So this is the precursor on red blood cell membranes, forming the basis for ABO blood group expression. So the type 2 precursor substance is modified by the H gene or the FUT1 to create the H antigen, which is further modified to form A and B antigens. So the takeaways for this image, so type 1 precursor is found in the secretions, saliva, and as well as body fluids, while type 2 precursor is found on the red blood cells and is responsible for ABO antigen formation. And then the glycosidic linkage determines whether a precursor functions in the body fluids or on the RBCs. So please take note that both precursors must be modified by the enzymes or the glycosyl transferases to express the ABO antigens.